All right, welcome to The Explainer. Today, we are heading to the most exclusive club in the entire universe, your own cells. We're going to pull back that velvet rope and take a look at the incredibly sophisticated security system that decides who gets in, who gets kicked out, and how the whole party inside keeps going.
I mean, have you ever really stopped to think about it? It seems so simple, right? But the answer is this amazing drama of physics and biology. And it all comes down to a super selective gatekeeper, a bouncer that is way more discerning and a whole lot more important than any you've ever seen at a nightclub.
So let's meet that bouncer. It's called the plasma membrane. Now, this isn't just some boring wall. It's an intelligent, semi-permeable barrier. It has a very, very strict door policy. And there are basically two ways to get past it, each with its own set of rules.
So you've got two main options here. On one hand, you have passive transport. This is the easy route. You're just going with the flow of the crowd, no energy required. But on the other hand, you have active transport. Now, this is like fighting your way against the crowd to get to that VIP section. And yeah, it's going to cost you some energy. Let's start with the easy way in, shall we?
Okay, so this is the general admission line. It's for all the molecules that don't need some special invitation. They just follow this natural principle called the concentration gradient, which basically means moving from where it's super crowded to where there's more space. Simple as that.
The absolute easiest way in is what's called simple diffusion. Think of this like being on the guest list. If you're a small, lipid-soluble molecule, you're lipophilic, which is just a fancy way of saying you're fat-loving. And because the membrane itself is made of fat, these molecules can just dissolve right through it. They slip past the bouncer, no questions asked. But what happens if you're not on that list? Well, what about all those water-soluble molecules and ions? They can't just dissolve through that fatty membrane. Nope, they need an escort. And that's where facilitated diffusion comes in. You can think of this as a really helpful bouncer, a special protein that recognizes you, grabs you, literally changes its own shape to open up a path, and then ushers you inside. The best part? Still no energy spent. But you absolutely need that specific protein to vouch for you. This is crucial for things like glucose to get into our cells and, give us energy. Okay, so that was the easy stuff. Now let's talk about the VIPs. These are the molecules that need to move against the crowd. They need to go from an area where there aren't many of them to a place where it's already packed. This is totally unnatural, and to make it happen, it's going to require a serious energy payment. And that's really the heart of active transport. You are fighting the natural flow, kind of like trying to push a giant boulder uphill. And that fight requires power. The cell has two main ways of paying the energy price to get this exclusive VIP access.
First up, we have primary active transport. This is like walking up and paying the bouncer directly with cold, hard cash. And the cash in this case is a molecule called ATP, adenosine triphosphate. It's the main energy currency of the cell. The energy from breaking down ATP is used to directly power a protein pump.
And the most famous bouncer that uses this direct payment method is a sodium-potassium pump. This thing isn't just any old protein. It is the head of security, and it is working at 24/7 in nearly every single one of your cells. It performs this incredibly precise, energy-driven dance just to maintain order. What's so cool is the rhythm of this whole thing. You can see it laid out right here. It all starts with 3 sodium ions from inside the cell that come and bind to the pump. Then boom, ATP pays the price. That energy literally changes the pump's shape, and it opens up to release those 3 sodium ions outside the cell. Then from the outside, 2 potassium ions hop on, the pump changes back, and releases them inside. This constant 3 out, 2 in exchange is so important. It creates this powerful electrical difference across the membrane. It's basically charging a tiny cellular battery.
Okay, so the second way to pay is a bit more clever. It's called secondary active transport. Instead of spending that ATP cash directly, this process hijacks the energy that's already stored in that gradient we just built. It's kind of like using the momentum of one VIP rushing through the door to sneak another person in right along with them.
And this comes in 2 main styles. You've got symport, which is like the buddy system. A molecule like sodium flows down its gradient back into the cell, and it literally drags another molecule, like glucose, along for the ride. Then you have antiport, which is more like a revolving door. As one substance moves in, another one gets pushed out in the opposite direction. Pretty smart, right? So we've covered individuals, small groups, and the VIPs. But what happens when the cell needs to move some really huge cargo?
I'm talking entire macromolecules like proteins or even whole bacteria. You can't just shove that through a little protein door. No. For this, you gotta call in the party bus. This whole process is called endocytosis. Here, instead of something passing through the membrane itself does all the work. It literally engulfs the material from the outside, wraps all the way around it, and then pinches off to form a little bubble called a vesicle inside the cell. This process has two main flavors. First up is pinocytosis, which literally means cell drinking. This is how the cell takes in fluids and other dissolved molecules. As you can see here, it's a three-step dance. First, molecules attach to the surface, then the membrane starts to fold inward, powered by these tiny muscle-like filaments, and finally it pinches off completely, forming that little vesicle inside. And of course, what comes in must eventually go out. Exocytosis is the exact reverse. It's how the cell takes out the trash, or how it secretes really important stuff like hormones. A vesicle full of material just travels to the membrane, merges with it, and bam, releases its contents to the outside world. Okay, we've seen how the club works. We've got general admission, the VIPs, the party buses. It's a pretty cool analogy, but what's the real-world importance here? Why should we care about these microscopic bouncers and transport systems? Because the answer, and this is the part that is just so incredible, is that these microscopic little processes are the absolute foundation for pretty much everything your body does every single second of every single day. So, seriously, why does this matter? Well, think about this. That sodium-potassium pump, our head of security, it is powering every single thought you have and every muscle you move by creating those tiny electrical signals. That buddy system we talked about, that's how your body absorbs nutrients from your food. And those party buses, that's how your immune cells literally eat bacteria to fight off infections. Every single one of these mechanisms is absolutely fundamental to you being alive.
And that just illustrates the whole point so perfectly. The plasma membrane is one of the most elegant structures in all of biology. It's not a wall, it's a dynamic, intelligent gatekeeper. Every second, trillions of these bouncers are making decisions, burning energy, and maintaining the delicate balance that keeps you alive and functioning.
It really makes you think, doesn't it? We've spent this time looking at the bouncers working the door of the cell club, but that just raises an even bigger question. What about the managers, the DJs, the bartenders? What other invisible, perfectly managed systems are running the entire party inside you right now, completely without you even noticing? It's a whole universe within.